Assalamualaikum warahmatullahi wabarakatuh. We are from Group 2. We are going to present our lab 4, which is trade dryer experiment.
For the first one is abstract. Tray dryers are the most basic and least expensive dryer type which operates by blowing hot air over the surface of a wet solid that is across trays arranged in the racks. Drying is a process of vaporizing and removing water or other liquids from a solution or solid liquid mixture to form a dry solid that involves simultaneous heat and mass transfer. This experiment has three objectives. The first one is to determine the drying rate and moisture content of the sample through removal of water. Next is to evaluate the different types of samples on drying rates. And the last one is to study the effect of velocity of air on drying rate of the sample by performing drying process. The grape is weighted and recorded the initial mass. The grape is placed on a tray, then slide into the drying oven. During the experiment, the mass of grape is recorded every 5 minutes until the grape observes a consistent mass for 4 consecutive times. Then repeat the same procedure using the shallow. The moisture content and the drying rate decreases over the time. The objectives are successfully obtained. Therefore, the experiment is successfully done. So now we move to the introduction. What is drying? Drying is a process of vaporizing and removing water or other liquids from a solution, suspension, or other solid liquid mixture to form a dry solid that involves simultaneous heat and mass transfer. Heat and mass transfer are critical aspects of drying process. Heat is transferred to the product causing the liquid to evaporate, while the mass is transferred as a vapor in the surrounding gas. For your information, drying is a crucial step in the manufacturing process of chemical industry and pharmaceutical industry. There are numerous drying methods, which are hot air application, contact drying, dielectric drying, freezing drying, and supercritical drying. These drying methods employ a variety of drying equipment, which are the vacuum shelf indirect dryers, continuous tunnel dryers, rotary dryers, drum dryers, and spray dryers. Tray dryers are commonly used in many industrial applications. The dryers are made of a tray that is held in a cabinet that is connected to a heat air source. The mechanism of tray dryers is the heat is channeled across the trays by an air current by conduction from heated trays or by radiation from heated surfaces. The tray must fit snugly in the cabinet to allow air to develop by passing the material to be dried. Tray dryers require more time to dry. Tray dryers are used to dry solid for around 12 to 48 hours. This is due to inexpensive capital and maintenance costs of tray dryers, but it have poor control and potential for producing more variable product quality.
For theory part, the tray dryer is an enclosed in an insulated chamber. It consists of trays which are placed on top of each other in the trolleys. So this tray dryer function is for drying and heating process. In industries, the tray dryer is the most widely used in food and pharmaceutical industries since the heating and drying process are really taking parts in both industries. After that, the tray dryer operates by passing the hot air over the surface of a wet solid on the trays. That means the wet solid will face dehydration by exposing it to a source of hot dry air until it dries enough to store at the ambient temperature and at the minimal spoilage. Other than that, this process involves heat and mass transfer. The heat and mass transfer are very crucial in this process. Why? Because the heat will be supplied to evaporate the liquid contained in the wet solids, and then the mass is transferred as a vapor into the surrounding gas. After that, we move to the next slide. So for this next slide, we can see the graph. We have two graphs here, which is the moisture content versus time and the rate of drying versus moisture content. The solids drying generally follow two distinct drying zones, which is constant rate period and falling rate period. So both of the rate period are separated by a breaking point called the critical moisture content. So if we see at graph A, point AB is referred to the constant rate period and point BC is referred to the falling rate period. So for point AB indicates the evaporation of the moisture from the saturated surface at a rate which is controlled by diffusion from the surface through the air. Other than that, the liquid transported to the surface also must be at a rate sufficient in order to maintain the saturation during the constant rate period. And then the constant rate period also depends on the air temperature, humidity, and the speed of moisture to the surface. At point B, a break in the drying curve occurs and is known as the critical moisture content. Point BC is called the first falling rate period, as I have mentioned previously. And then as drying proceeds at point BC, moisture reaches the surface at a decreasing rate. And then other than that, the mechanism that control its transfer also will influence the rate of drying. At point BC, moisture reaches the surface at a decreasing rate. And then other than that, the mechanism that control its transfer also will influence the rate of drying. As the surface is no longer saturated, it will tend to rise above the wet bulb temperature and resulting in the point CD. So the point CD is the second falling rate period. Let's move to the objectives. So the first one is to perform drying process of two different types of sample by using selected drying equipment, and then to collect the weight of samples by using weighing scales. And the last one is to evaluate the effect of different types of samples on drying rates.
So, a grape and a shallot were chosen as the food samples or the main material of this experiment, and they will be labeled as sample A and sample B respectively. So, sample A is for grape and sample B is for the shallot. So, the apparatus used for this experiment is a tray, a drying oven, a digital weighing scale, as well as several sheets of paper towels. So as you can see, the samples chosen are comparable in size, whereby they have an approximate diameter of 2 centimeters. You see, sample A, which is the grape, is actually a juicy fruit which is ellipsoid in shape, and it has a very smooth surface or outer skin to cover up the juicy pulp on the inside. Sample B is actually like an onion, but it is smaller and more elongated in shape. It is bulbous and has a papery multi-layered skin, just like an onion. Now, in order to prepare the material, a paper towel is lightly wetted to clean the samples of any debris or residue, and then the samples will let to dry for 30 minutes at room temperature before undergoing the experiment. So as you can see, the samples chosen are comparable in size, whereby they have an approximate diameter of 2 centimeters. You see, sample A, which is the grape, is actually a juicy fruit which is ellipsoid in shape, and it has a very smooth surface or outer skin to cover up the juicy pulp on the inside. Sample B is actually like an onion, but it is smaller and more elongated in shape. It is bulbous and has a papery multi-layered skin, just like an onion. Now, in order to prepare the material, a paper towel is lightly wetted to clean the samples of any debris or residue, and then the samples will let to dry for 30 minutes at room temperature before undergoing the experiment. So as you can see, the samples chosen are comparable in size, whereby they have so the whole experimental procedure can easily be compressed into a simple process flow diagram. So the first thing that needs to be done is to preheat the oven until 200 degrees Celsius for 10 minutes, and then simply weigh and record the initial mass of the sample, and then you place the sample onto a tray, and then you slide into the oven to be dried, and then the sample needs to be weighed and the mass needs to be recorded every 5 minutes. The sample continues to be dried until it reaches a consistent mass for 4 consecutive times. And last but not least, just simply repeat the same procedure with the next sample, or in this case, sample B. Now, shown are the before and after pictures of sample A and sample B. So it can be observed that both samples have that wrinkly and shrunken appearance as a result of them being dehydrated of their moisture. And last but not least, this is a table of the data collected every 5 minutes and will be further discussed by another group member. Okay, so since we have obtained all the data and all the masses of sample A, sample B, we can determine the drying rate, which is the rate at which the samples are being dried, at which the moisture in the samples are being dried. So since the objective of this experiment is for us to determine the effects of the drying rates, therefore the drying rate is very crucial in this experiment. But in order to calculate the drying rate, we need to calculate the moisture content first, which is the quantity of water inside the sample. So how do we calculate it? It is pretty simple. We need to subtract the mass of the wet sample at that one particular time interval by the mass of the dried sample at the end of the experiment, and divide it by the mass of the dried sample at the end of the experiment as well. So the sample calculation is shown in the video. So once we've got the moisture content values and it has been tabulated as well, we can calculate the drying rate, which is also very simple. We just simply need to subtract the first moisture content at the very first interval and subtract it by the moisture content at the next time interval and divide it by the time duration. So you can simply repeat the same step, the same calculation for the next time duration and time interval. So the results will be tabulated in the video, and the results will be further discussed by another group member.
Hi, my name is Umar Hazim bin Muhammad Fauzi from Group 2, and I will be presenting the discussion of the experiment. In conducting this experiment, an oven was used as a drying equipment where temperature and time are the most crucial factors that affected the efficiency of the removal of moisture. The samples chosen for the experiment were grape and shallot to obtain a different result regarding the drying rate and moisture content. The comparable sizes and moisture content of grape and shallot are different in value, which gave a different result at the end of the experiment, respectively. Both samples went through the same drying process with the same amount of time and temperature, which is 200 degrees Celsius, and the mass was taken every 5 minutes. The experiment was conducted until both samples record consistent mass for 4 consecutive times. Based on the graph, for sample observation A, the mass recorded was 12 grams for 0 and 5 minutes, 11 grams for 10 minutes, 9 grams for 15 minutes, 8 grams for 20 minutes, 7 grams for 25 and 30 minutes, 5 grams for 35, 40, 45 minutes, 4 grams for 50 minutes, and 3 grams for 55, 60, 65, and 70 minutes. While for sample observation B, the mass recorded was 12 grams at 0, 5, 10 minutes, 11 grams at 15 and 20 minutes, 10 grams at 25 minutes, 9 grams at 30 minutes, 8 grams at 35, 40, 45, 50, 55, 60, 65, 70 minutes. From the graph, we could conclude that we got a proven result from the experiment, as drying did not occur in between some of the intervals of the graph due to consistent mass. There are differences in both samples, which is sample A is an ellipsoid shaped, which has smooth skin, and it is also a juicy fruit, while sample B is almost like an onion, but smaller and elongated in shape. Sample B is bulbous and also has a multi-layered peppery skin. Heat transfer and mass transfer are the two important aspects in drying process. Heat generated from the oven is transferred to the sample to evaporate liquid, and mass is transferred as vapor into the surrounding. The drying rate is determined by the set of the factors that affect heat and mass transfer. Drying of solids is commonly understood to follow two distinct drying zones, known as the constant rate period and the falling rate period. The two zones are divided by a breakpoint, called the critical moisture content. The control of moisture levels in solid materials is a vital process which is used in many industries to produce chemical products. For instance, in the pharmaceutical industry, drying normally occurs as a batch process where it is the key step to manufacture quality attributes of the final dosage form in terms of tableting and encapsulation. Lastly, during conducting the experiment, there are some precautionary steps that ought to be taken. Firstly, we must use a heat-insulated glove along the whole experiment to prevent any injuries due to the hot oven at 200 degrees Celsius. Other than that, the digital weighing scale used needs to be tested prior conducting the experiment to identify any faulty of the apparatus. Next, in order to obtain an accurate result, both sample A and sample B should be wiped a few times to make sure that all of the moisture on the surface are removed completely.
As a conclusion, this experiment has achieved the objective, which was to dry grapes and shallots using specific drying apparatus such as an oven, to determine the weight of grapes and shallots using digital scale, and to determine the effects of grapes and shallots on drying rates. From this experiment, both samples went through the same amount of drying process with the same amount of time until it reached the same mass 4 times consecutive and temperature 200 degrees Celsius respectively, and the mass was taken every 5 minutes. The result of the drying process performed on the grapes and shallots were consistent with the theory, which is the drying rate did not occur in between some of the interval of the graph due to consistency of mass. The experiment conducted was running smoothly except for a condition where the equipment was not well maintained. In fact, troubleshoot the experiment is important as it yields errors in results obtained. Therefore, since the objective of the experiment is successfully obtained, the experiment is successfully done. Even though the experiment was conducted successfully, there may be some errors in this experiment. Results can be improved. First of all, the student should be more alert on conducting the timer or the stopwatch to avoid the parallax error, which means we will get more accurate value of mass of value samples for every time taken that have been taken. The equipment of the apparatus such as an oven should be well isolated to prevent the heat from inside the oven to be released to the surroundings, which will resulting in slower drying rate. The next one is the oven and the weighing scale must undergo maintenance or check it before starting the experiment to prevent zero error. This action will prevent any faulty of the apparatus. Last but not least, the digital weighing scale needs to be replaced to avoid miscalculation. This is because our weighing scale is limited to 2 significant figures only. Thus, it will give a less accurate value of data. So, there are few references that we use to complete the experiment report.